higher as the human factor.
Thank you, Jerry. Thank you, everyone, for coming. And today, it's very nice to be here, and I have my very nice visit so far today. So today, I'm going to talk about temperature sensation in fly larvae. You know that animals like us can sense temperature. Temperature sensation is very important for animals to live noxious temperatures like this kangaroo. It has to live to escape from this noxious high temperature. And the penguins, they can sense the extreme cold and keep warm together. And this eagle, they just try to escape the wildfire. Animals also use their temperature sensation to look for their best environment. These animals and geese, they are trying to migrate thousands of miles every year to look for the better environment for them to survive. Sometimes animals use their thermosensory system to look for their prey, like this snake. It has an organ called pig organ on its face. Inside of pig organs, there are temperature responsive neurons. And the snake uses those cells to detect the temperature of their prey. So in dark, it cannot see this mouse, but it can read the temperature of this mouse. Unfortunately, sometimes we are the prey. Mosquitoes detect our temperature and bite us. Environmental temperature variation is particularly significant for insects. This is because they're small, they cannot control their temperature, so they must rely on the ambient temperature to set their body temperatures. Here, I would like to use Mexican jumping bean as an example to show you how in nature insects avoid bad temperatures. These are Mexican jumping bean. If you go to Amazon, you can get them. And they can jump because the mouse larva, the Cydia larva, stay inside. This is a very nice environment for them. And except that the extreme heat from the desert. And this heat can dry them, can kill them. And so they must escape this deadly heat. So they move, and that's caused the jump. And they want to find a piece of shade to cooling down. This is the experiment scientists did. Can see this side is the heat lamp, and this side is the ice pack. The Mexican jumping beans avoid the high temperature and go to the cold temperature. Unfortunately, we cannot study this mouse directly. We need a model. My lab used fruit fly as a model to study temperature sensation. You might know some general benefit to use fly as a model to do the research. They are easy to rear. They share 66% genes with human genome. They have short lifespan. And this is the Drosophila life cycle. We have male, female, they mate together. The female give us the embryos after a day, first instar larva come, and another day, second instar larva, another day, third instar. And the third instar will last about 3 days and then go to the pupal stage and go to the adult. This process at 25 degrees Celsius will last about 10 days. Temperatures significantly affect the development of Drosophila. So at 25 degrees Celsius, this process is 10 days. At 22, it's about 14 days. And at 18 degrees, it's about 3 weeks. And fruit fly is a model, it's a good model because it has powerful genetic tools. There are many great, powerful genetic tools in flies. And if I have time, I will introduce you one of them we developed recently in the end. But if you can only know one of them, that must be the GAL4 UAS system. So GAL4 is a yeast transcription activator. It is under control of tissue-specific promoter or enhancer. So the GAL4 line determines where to express. After GAL4 is expressed, it binds a DNA sequence called UAS, and then it will trigger the expression of the gene downstream UAS. In this case, it's gene X. So the UAS line determines what to express. Okay, last but not least, Drosophila is particularly good for temperature sensation because it gives us robust behavior responses to temperature changes. This is the behavior we use. So this is the temperature gradient. The left side is cool temperature. It's about 13 degrees Celsius. The right side is the warm temperature, and it's about 31 degrees Celsius. We give larvae about 10 minutes to explore their favorite temperatures. You can see just they move up and down, left and right to find their optimal temperatures. After 10 minutes, we will take a picture and count in each temperature interval how many larvae are there. And after that, we got this quantification figure. The x-axis is the temperature, y is the fraction of larvae within each interval. You can see between 23 and 25, there are largest number of fly larvae. They suggest that fly larvae prefer this temperature. Then we really want to know what cells guide larvae choose 24 degrees Celsius. This is the fly larvae. This is the anterior part of the larvae. We can see these circles. Each circle represents a pocket-like structure. In each of these structures, there are dozens of neurons. Most of them are the chemosensory neurons. We focus on this orange circle. We call them DOG, the dorsal organ ganglions. In each dorsal organ ganglions, there are about 30 neurons, and most of them are olfactory sensory neurons. So their function is for olfaction or smell. And among them, there are 5 temperature responsive neurons. 3 of them are the cool cells. You can see these three blue cells. And 2 of them are warm cells. Cool cells, they name as cool cells because they are activated by cooling, and warm cells are activated by warming. Here is the calcium imaging to show you how cool cells and warm cells respond to temperature changes. This corner gives you the time and the temperature. The whole video will last about 2.5 minutes, and you can see the time. And during the first 30 seconds, the temperature will now change, and then temperature will decrease, increase, decrease, increase. And calcium imaging, you might know, it uses calcium indicator to estimate the neuronal activity. So when the fluorescence is high, those 2 neurons are activated. That means neurons are activated. When the fluorescence level is low, that means neurons are deactivated. So right now it's about 27 degrees Celsius. It's a high temperature in this experiment. These 2 neurons are warm cells. And now I'm going to show you the video. And during the video, you are going to see 3 neurons come out and disappeared according to this temperature change. So here is the video. You can see cooling down and the 3 neurons come out and then warming up. And let's slow down this video. So this is the temperature at 27 degrees Celsius. And then when the temperature go down to here, there are 3 neurons come out. 1, 2, 3. These 3 neurons are activated during cooling, so they are cool cells. At the same time, I hope you can notice these 2 cells, they are warm cells, and they are deactivated when temperature decrease. And when temperature increase again, and to go back to the room temperature, even now, just 20 degrees, can see cool cells disappear and warm cell come back. This is the quantification result of cool cells. You can see the lower panel is the temperature. The first 30 seconds temperature did not change, and then decrease, increase, decrease, increase. The upper panel show you the temperature change. we can see that the response did not change much. And then during temperature decrease, the response increase. This suggests cool cells are activated by cooling. This is the response of warm cells. Again, they are activated by warming. I hope you can focus on from the 60 seconds to 90 seconds. When temperature increase, and you can see those warm cells response increase, suggests that warm cells are activated by warming. So now we know that cool cells are activated by cooling, warm cells are activated by warming. Our next question is, what molecules guide these temperature responses? These molecules are named inotropic receptors on IRs. A brief introduction about inotropic receptors. They are inotropic glutamate receptors. They're inotropic glutamate receptor variant, I should say this. Inotropic glutamate receptors including AMPA receptor, NMDA receptor, kinase receptor. Usually, 4 of them form a heterotetramer, and each of them has this structure. It has an N-terminal domain, the ATD domain, the ligand binding domain, LBD domain, the transmembrane domain, TMD domain, and the C-terminal domain, CTD domain. IRs can be divided into co-receptor and tuning receptors. And the co-receptors have similar structure as inotropic glutamate receptors. They have ATD domain, LBD domain, TMD domain, and CTD domain. Co-receptor pair with different tuning receptors to respond to different stimuli. On the other hand, the tuning receptors, they usually do not have typical ATD domain, and they must pair with 1 or 2 co-receptors to respond to specific environmental stimuli. In cool cells, the tuning receptor is IR21A. It pairs with IR25A and IR93A to form the cool receptor. And in warm receptors, the co-receptors are same. They are IR25A and 93A, but the tuning receptor is IR68A. So this is our staining result of IR68A, IR21A, and IR93A. For the 68A, they express 2 cells in each dorsal organ ganglion. These 2 cells are warm cells. IR21A, they express 1, 2, 3 cells in each dorsal organ ganglion. These 3 cells are warm cells. IR93A is a co-receptor. It expressed in 5 cells. 3 of them, you can see 3 of them are co-localized with IR21A. They are cool cells, and 2 of them co-localized with IR68A. They are warm cells. So now we need to check whether these IRs are necessary for the temperature responsiveness in these warm and cool cells. So as I mentioned, this is cool cells response to temperature change. They are activated while temperature decrease. See, temperature decrease, they activated. When we mutate IR25A, IR93A, and IR21A, this cooling response largely diminished. And such cooling responses can be rescued by expressing the respective IRs into cool cells. So this suggests that these 3 IRs are necessary for cooling responses in cool cells. And then we ask a key question. This question is whether these 3 IRs can give cooling sensitivity to other cells. This is key because this question will directly answer whether these 3 IRs form the cool receptor or not. We use heating cell as the model to study this question. Based on the name, you know that heating cells are activated by high temperature. They locate at the base of a raster. At the base of a raster, there are only 6 neurons. 3 of them are heating cells and 3 of them are cooling cells. So here is the activation of the heating cells. you can see that they are activated during warming and deactivate during cooling. Warming, they activate, cooling down, they dim down, and they activate again and cooling down. Okay, so this is our quantification result. We can see that these cells are activated during warming, deactivate during cooling. These blue arrows show you the lowest temperature point, so we cannot observe any activation. Then we go to our question, if we express these three IRs, IR25A, IR93A, and IR21A into heating cells, can these three IRs give cooling responsiveness to heating cells? Luckily, the heating cells have endogenous IR25A and IR93A. So we only need to express IR21A into heating cells. And then the question becomes, can IR21A confer cooling responsiveness upon heating cells? Again, we use calcium imaging to answer this question. During this calcium imaging, the temperature will increase first and then decrease, and then increase, decrease. These three white arrows show you where the heating cells are. So you can have a look. So right now, temperature did not change and start to warming up. They light up, and cooling down, they dim down first and then light up. And then warming up, they light up again, and cooling down, they dim down first and light up. So we give this cycle 3 times. They very well behave like they are activated during all 3 cycles. This is the quantification result. These are heating cells. So they activated during temperature increase. So you can see temperature increase, they activated. And then during temperature decrease, we can see another activation. This result suggests that IR21A confers cooling sensitivity to heating cells. Then we ask whether this cooling sensitivity requires 25A and 93A. Yes, they do, because in the IR25A mutant, we cannot observe such cooling responsiveness. So now we know that IR25A, IR93A together with the tuning receptor IR21A form the cool receptor in cool cells. And how about warm cells? And the IR68A, 25A, 93A, are they necessary for warm cell response? This is true. This is the data from Dr. Samuel's lab published in 2021 in Science Advance. And you can see the first panel, the upper panel is temperature oscillation. The temperature increase first and then decrease. And the middle panel is the wild-type response. They are activated by warming and deactivated by cooling. And the lower panel is the mutant. When IR68A, IR93A, and IR25A are mutated, the warming response disappears. This suggests these 3 IRs are necessary for warming responses in warm cells. Okay, now we know these IRs, IR21A, 25A, 93A together form the cool receptor. IR68A, 25A, 93A together form the warm receptor. We want to ask how these cool and warm cells and their IRs guide larvae to choose 24 degrees Celsius. So this is the behavior from the wild-type larvae. They prefer 24 degrees Celsius. And when IR21A is mutated, they prefer 18 degrees Celsius. And when IR68A is mutated, they prefer a warmer temperature. So this result tells us in 21A mutant, the cool cell do not have functional cool receptors. So cool cell cannot respond to cool temperature, and they cannot guide larvae to leave the cool region. That's why the larvae stay in a cooler area. And similarly, in IR68A, warm cells do not have their functional warm receptors, and they cannot guide larvae to leave warmer region. So more larvae choose the stay at the warm region. And this is our model. The cool cell guide larvae leave cool region, warm cell guide larvae to leave warm area, and at about 24 degrees, a balance forms, and so the larvae stay at about 24 degrees Celsius. And this is the first summary of today's talk, that is cool and warm cells and their IR receptors together guide fly larvae to about 24 degrees Celsius. Now I would like to move to the second part of my talk. If we go back to look at this life cycle, we know that first larval lasts about 1 day, second larval lasts about another day, and the third one, they last about 3 days. So we further divide the third larvae into the third instar larval into the early third instar, mid third instar, and the late third instar. Early third instar are about 3-day-old larvae, mid third instar about 4-day-old larvae, and the late third instar are about 5-day-old larvae. Just now I told you larvae prefer 24 degrees Celsius, and this 24 is there from first instar to their early third instar. The data we collected, I showed you, are from the early third instar. We and others together also showed that first, second instar larvae also prefer 24 degrees Celsius. Now let's move to a different larval stage, that is late third instar. Late third instar larvae are about 5 days old, and they are the last larval stage before they go to their immobile stage, the pupal stage. When we run the behavior, surprisingly, they prefer a much cooler temperature. They prefer about 18 degrees Celsius. And then we want to ask why. What is the mechanism cause this transition? And we go back to check our cool receptor in cool cells. And we check IR25A, IR93A, and IR21A. Here is the example to show you IR21A. We can see that in 3-day-old larvae, the IR21A in their cell body, we can see them, although not very strong, but they are very strongly, highly enriched in their dendrite. But when larvae grow to 5 days old, their cell body signal dramatically decrease. We almost cannot see them, and their dendrites, they are very faint too. So this result suggests when larvae grow up to 5 days old, their IR expression significantly decreased. And then because their IR level decrease, we propose that their cooling response should decrease too, right? And then we use calcium imaging to check the cooling response in cool cells. So this is 3-day-old larvae, and at 26 degrees Celsius, this is their fluorescence level, which is pretty low, suggests that they are not activated. When temperature decreased to 14, we can see significant increase of their fluorescence level, suggest at cool temperature, these cool cells are activated. This is what I showed you just now, similar result. However, when the larvae grow up to 5 days old, you can see that at 26 degrees Celsius, that is a warm temperature, the signal level probably similar to the 3-day-old larvae, but they cannot increase when temperature decreases to 14 degrees. So this result clearly show that the cooling responses of cool cells also decrease. Then we go to our behavior data. Because our data show you that IR25A, IR93A, IR21A level decrease in cool cells when larvae at 5 days old. So that suggests the cool cell cannot respond to cool temperature. So they have no ability to guide larvae to avoid or to leave this cool temperature. So they have to stay here. If we compare the 5-day-old wild type data versus the 3-day-old IR21A mutant data, they look very similar. Okay, so if our hypothesis is correct, then we should be able to reverse this phenotype, right? What we did is express IR25A, 93, and 21A in cool cells and examine the 5-day-old behavior again. This is our result. You can see that one larvae express IR25A, 93A, 21A in cool cells. This larvae prefer a warmer temperature. So this result suggests the expression of these IRs changes temperature preference and show you decreased expression of IR receptors in cool cells results in animals stay at cool region during late larval stages. Now I would like to move to the third part of my talk today, and let's go back to look at our calcium imaging result. Remember, these two cells are warm cells. They are deactivated during temperature cooling. And if we look at our calcium, there are two cool cells come first and the third one come later. And let's slow down. So if we see here that we can see two of them, I will slow it down. Okay, so you can see two of them come and then the third one come out. The first two, they pair with warm receptors, and we call them A-type warm cells. And the third one, they do not pair with A-type. They do not pair with the warm cells, so we call it B-type. This is the schematic diagram to show you A-type cool cells versus B-type cool cells. So these two pair with warm cells are A-type cool cells, and this one does not pair with warm cells. It's B-type cool cells. So my lab used 20% sucrose to collect larvae from their food, and when the larvae stay in the 20% sucrose for about 1 hour, we can see the robust response of both A-type and B-type cool cells. You can see that their response are similar and robust. However, when these larvae stay in 20% sucrose longer, like such as 3 hours, we can see that A-type response still robust. They can maintain their response. However, B-type response significantly decrease. So this suggests the 20% sucrose decreases B-type cooling responses. And you know that 20% sucrose is a type of hypertonic solution. So it's in this kind of solution, when the cell inside of it, the water will leave the cell, right? Then our question is the phenomenon we observed is sucrose specific or other hypertonic solutions can also cause similar result? And to answer this question, we change a hypertonic solution. In this case, we use 0.5 mole sodium chloride. So we can see that this is temperature, and within 1 hour incubation in 5 mole sodium chloride, and both A-type and B-type strongly respond to temperature change. And B-type in this case actually responds stronger, and we don't know the reason here. But after 3-hour incubation, the A-type maintain their response, and the B-type response significantly decrease. Then, of course, we want to ask why. Why hypertonic conditions have selective impact on B-type cool cells, but do not affect A-type cool cells? We go back to visit our immunostaining, and what we found here is that if we look at our, okay, first of all, this is the B-type because it did not pair with the warm cells. R68A GFP label warm cells, and these two cool cells are A-type cool cells because they pair with the warm cells. And this one is the B-type cool cells because it does not pair with warm cells. So first of all, let's look at IR21A. If we compare the level of IR21A in A-type cool cells and B-type cool cells, their level are similar, and this is the quantification data. And the quantification is the A-type versus B-type. If it is close to 1, that means that they have similar expression. And similarly, IR25A expression in both A-type and B-type are similar. This is the quantification. However, if you look at IR93A, their expression level in A-type and B-type are significantly different. In A-type, there are much more IR93A than in B-type. And we can see that this level is much higher than 1, significantly higher than 1, suggests that A-type cool cells express higher level of IR93A than B-type cool cells. Then our question is, does reduced expression of IR93A increase the vulnerability of B-type cool cells to hypertonic conditions? To answer this question, what we did is trying to increase the B-type IR93A by overexpression IR93A in cool cells. And luckily, we did make the IR93A level in both A-type and B-type are similar. You can see here that these two yellow arrows show A-type, and this what's the name? The white arrowhead show B-type, and this is similar. And you can see that A-type and B-type have similar amount of IR93A. Quantification confirmed our result. So we use this larvae, and then to do the calcium imaging, we found that when we expose this larvae to 20% sucrose, their B-type response recover. So this result suggests that the B-type cool cells with reduced IR93 expression function as an integrator for thermal and tonic informations. So then you probably want to ask me, hey, Lina, why do you bother to study these IRs? They are not even expressed in humans, not in mammals. Okay, so first of all, these IRs are conserved across protostomia. So temperature responsive IRs, including those IR25A, IR93A, IR21A, IR68A, they are among the most conserved IRs. And the function of these IRs has been studied in mosquitoes. They not only have conserved function, but they guide the mosquito host-seeking behaviors. So without these IRs, the host-seeking behaviors is disrupted. So because they are not expressed in us, they provide a potential target for developing novel inventions to mitigate the spread of insect vectors of disease and reduce their blood feeding behaviors. Make sense? Okay, so now we still have time. So I would like to spend the last 5-10 minutes to introduce you a genetic tool we developed recently and published this year. And I will not go into the data, and I didn't prepare to go into the data. I will just give you a brief introduction of this tool. This tool is named FlipSort. FlipSort is the functional labeling of individualized postsynaptic neurons using optogenetic and transtangle. So FlipSort, first of all, use transtangle to access postsynaptic neurons genetically. If you are not familiar with transtangle, transtangle is a very beautiful genetic tool. It allows you to use the presynaptic GAL4 line to label your postsynaptic neurons. So in most cases, it's very hard to label postsynaptic neurons of the specific presynaptic neuron, right? It allows you to label them without driver line. So this is the beauty of the transtangle. And the second, the flip-stop uses optogenetic approaches to activate or inhibit postsynaptic neurons in a random and sparse manner. So activate neuron, we use CS crimson. CS crimson is channel rhodopsin. It is a cation channel. When the red light is on, the CS crimson will be activated, and then cation will flux into the neuron to activate the neuron. To inhibit postsynaptic neuron, we use GTACR2. GTACR2 is the chloride channel. When the blue light is on, and it will allow the chloride to flux into the cell, so it will hyperpolarize the cell or say inhibit the cell. And to achieve a random and sparse manner, what we use is the FRT stop FRT cassette and heat shock promoter controlled FLP. I will go over that in detail in next slide. And the last flip-stop has the fluorescence markers tagged with optogenetic genes to visualize these neurons. Okay, so they have three things. And now it is the schematic representation of the flip-stop. This is presynaptic neuron. These are postsynaptic neurons. So as I mentioned, in presynaptic neuron, you need a driver line. You need a GAL4 line. And as long as you have this GAL4 line, the GAL4 will bind the UAS of the ligand. This ligand is transtangle ligand. So you can imagine that after the GAL4 binds the UAS, and then this ligand will be expressed. When this ligand is expressed, it will go to presynaptic terminal, and there they will bind to the transtangle receptor there. And after this binding, it will recruit the protease to go there and to free this QF. QF is like the GAL4. It's a transcription activator. So when the QF is freed from the membrane, it goes to the nucleus and binds to a DNA sequence called QUAS. And this binding will allow the downstream reporter to be expressed. If we only stop here, just only have this part, that is the classic transtangle. What we did is to add this part. We add a QUS, this triangle are the FRT site. QUS, FRT, stop FRT sequence, then followed by CHR. CHR is channel rhodopsin. And after channel rhodopsin, there is the fluorescence tag. And also include this heat shock controlled FLP. So after heat shock is introduced, the heat shock here, the promoter will guide the expression of FLP, you can see here, and then the FLP will be expressed. You might know that FLP can cause the recombination of two FRT sites, and this recombination will allow the remove of the stop sequence, and so the channel rhodopsin will be expressed. So this channel rhodopsin is either CS crimson or the GTACR. CS crimson, again, it is CS crimson is again it's trying to activate neuron, and the GTACR function to inhibit the neuron. At their C terminal, we have a fluorescence tag to allow us to visualize which neurons express the GTACR on CS crimson. And the other thing I would like to say is that this heat shock, as I mentioned in the previous slide, like this slide, we use this to achieve the random and sparse manner. How we do that is to play with the heat shock. We can play that heat shock time, just how long you do the heat shock, and also the heat shock temperature, so that we can make the FLP to be expressed in certain number of neurons, but not all of them. So to achieve that, the sparse labeling. Okay, so if we want to identify the necessary neurons necessary postsynaptic neurons for certain behavior, we should use flip-flop I. Flip-flop I include, you don't need to worry about this HC. HC is just the HC GAL4, it's presynaptic neuron, a presynaptic driver. So we have the flip-flop I, and flip-flop I include this HSFLP, heat shock controlled FLP, and we have UAS transtangle, that is the UAS transtangle ligand, and also their what's that receptor. And then QUS FRT stop FRT GTACR2, which is tagged with a YIP, and also another QUS MTD tomato. This is the fluorescence reporter. Okay, to use this tool, you need a presynaptic driver. And after you cross them together, what you need to do is to heat shock first. And heat shock, the function of heat shock is to drive the expression of FLP, see this one, and then the FLP will drive the recombination between this FRT site. So the stop sequence will be removed. And after that is removed, the GTACR YIP will be expressed. And then this one doesn't need the heat shock. It will be expressed as long as the transtangle work. And then you need to incubate these flies in 18 degrees for a certain amount of time. It depends on how strong your presynaptic GAL4 is, and also the GTA's flip-flop I and A use different time. So in this case, it's about 7 to 8 days. And you might ask why it's 18 degrees. This is the limitation of transtangle. The best development time to develop transtangle needs 18 degrees. And 2 days before your behavior assay, you need to add ATR into your food. This is to support the function of GTACR on CS crimson. And after that, 2 days later, you run your behavior. In this case, it requires a single fly behavior assay, because you will see this one is my control, like under ambient temperature, and under ambient light. And this one is the real assay under blue light. And so you should see the wild type behavior under the room light and the behavior you want to see under the blue light. And after that, you pick those fly that show the behavior you want to see and dissect their brain and check which neurons express those GTACR. Among those neurons, some of them are necessary, but of course there are some other neurons also express the GTACR. So you need to do more than one. We did 30, and to find those neurons that express in all of them, that are the neurons necessary for this behavior. So similarly, you can use flip-flop A to identify postsynaptic neurons that are sufficient for certain behavior. Again, the flip-flop A includes this heat shock controlled FLP, the transtangle, FRT stop FRT, and in this case is CS crimson, because CS crimson drives the activation of neurons, and it is tagged with M-cherry. Because this tag is M-cherry, so we use the GIP as the reporter. And again, you need heat shock, you need ATR to support the function of the CS crimson. And in this case, you need red light to activate your neurons. It's here, right? And then if you find the fly behave what you want to see, you need to collect those flies and to dissect their brain, and you can identify the postsynaptic neurons that are sufficient for certain behavior. So flip-flop is used to identify postsynaptic neurons that are necessary and sufficient for certain behaviors. This is my last slide, and I would like to thank my lab. Ali was my first PhD student. She was the one to develop flip-flop. And the behavior and the calcium imaging data I showed you is largely generated by Hua, by Alisa and Jordan. Alisa and Jordan are two very creative, very amazing lab manager working in my lab. And one lab to Jordan went to the medical school. Alisa went to PhD program. And Hua is my research scientist. These are my funding resources. Last but not least, I would like to thank my flies who give me the very nice, robust behavior. So thank you very much, and I'm happy to take questions. Thank you. I don't know whether that's true or not, but I have a hypothesis. I never really know whether this is true or not. We think like for that is the 5-day-old larvae, that's late third instar. And the next stage, they have to stay there for 4 days, and they cannot escape anything, right? It's basically, so they need to find a safer place. For them, cooler is safer than warmer, I think. Yeah. Because that's the because the temperature, if you think about 25 degrees, their favorite temperature, right? And when the temperature go up to like 29, they will start sterile, and go to like higher, they will die. And but if they can decrease to like 18, they will be totally fine. And even as he mentioned, like 15, they will be okay too. So that is safer. Okay, so for your first question, do I want to check what gene upregulate, deregulate between the 3-day and the 5-day-old larvae? I actually did the RNA-seq, and I didn't find very promising genes and to do this to continue to study. And one suggestion is just hormone change, because they are ready for the pupal stage, and their hormone regulation change. And that's probably the good angle to study. We didn't start yet, but that will be, if I need to study, I probably will start from that point. And for the second is whether when I express the IR21A in heating cells, I checked the calcium imaging, and did I check whether I checked their behavior? The answer is no, we didn't check the behavior. And so that's a good question. Actually, I should check that, because in adult, that is adult heating cells is in adult, and in adult, those 3 heating cells drive the avoidance in their 2-choice assay. You probably know the fly maze, and the 2-choice, one side is 25, the other side is 31. And if you do not have those 3 cells, they will not be able to tell this is 25, this is 31. They have no preference. Those 3 cells functions to guide larvae to avoid that 31 degrees Celsius. And it will be cool to see what will happen, like even cooler, the 25A versus 18, maybe it may change the behavior. That's a good suggestion. We probably will check. Yeah, thank you. So you mean the other environmental stimuli, like the light condition? We actually did the experiment in pretty dark. That's really depending on the who did the experiment. Like if I do the experiment, I like to do the one key thing is that because they have phototaxis, you have to maintain the constant light, right? The light level must be same on both sides. And we know that if you have maintained the constant light and they have the similar behavior, just that they all like the 24 degrees Celsius. But we noticed one thing is that in the early morning, they kind of prefer a little cooler, and the later afternoon, not later, just midday and maybe early afternoon, they prefer a warmer temperature. And this is also true for the adult fly. And Dr. Famiga Hamada have the current biology paper and to show the temperature preference shift in adult flies. Yeah.
Actually, during development, the activity
Yes.
expression,
No, expression decreased. Yeah. Yes.
what people
We don't know. We really don't know that. But that is when we go to the eLife, we finally published that in PLOS Genetics. That was one of the suggestions from the eLife reviewer to check the hormone, whether those hormone change regulate that change. The reason we didn't do that is because even we finished that, they will consider it as a new submission. Just we did all the experiment and still a new submission, so we directly go to the PLOS Genetics. Yes.
So we sort of touched on this when we were talking in my office. We care to speculate on how integration happens downstream of these.
We do want to see, and in adult, they have already tried to check the, as I mentioned in the Arista, there are 6 neurons, only 6 neurons, right? 3 heating, 3 warming, and they already, 3 heating, 3 cooling. They already try to understand the warming cell, that's the heating cells, go to the VP2. If you don't know, just they go to the antennal lobe, and the antennal lobe can be divided into different glomeruli. So the heating cells go to VP2, cooling cells go to VP3, and they also have the sacculus. Sacculus has their moist cells, dry cells, and also another heating cells, and they go to VP1, VP4, and VP5. So in total, there are 5 glomeruli regulate the humidity, the temperature, and people already start to understand they have projection neurons. Projection neurons receive information from glomeruli to higher level of the brain, such as mushroom body, like the lateral horn. And they do have those projection neurons can receive information both from VP2 and VP3. That's basically the heating information and cooling information, and they can be activated by both and then sent to the higher level. And in larvae, I don't believe there are any information has been published about the next level of the integration. But one thing is super interesting, and I cannot explain, is that you can see that in larvae, in each dorsal organ ganglions, there are 3 cool cells and 2 warm cells. And those 3 cool cells go to the same glomeruli, but those 2 cells each go